of Welcome to our second day of the annual meeting of the Mountain West Clinical Translational Research Infrastructure Network. Today, I think yesterday we have a very successful meeting with about 64 participants. So today, hopefully we'll have another successful meeting. We have 8 presentations this afternoon. And so I'll go ahead and let Curtis Noonan and Scott to introduce the speakers.
Okay, great. Thanks very much. It was a great day yesterday, and the speakers did a fantastic job of keeping us all on time. So that was much appreciated. As you know from the agenda, today we're going to have 8 presentations and a couple of breaks worked in there during the afternoon, short breaks. Scott Seville and I will be trading off on moderating duties. So I'm going to start with the first session here, which is begins with Jim Becker. And Jim, do you have you can go ahead and share your slides if you're ready to do that. Okay. So Dr. Becker is, and just to remind everyone, we'll have 15-minute presentations followed by 5-minute question and answer. It's helpful to input any questions in the chat window as well as we're going along. And oftentimes, as we noted yesterday, there's not enough time to answer all the questions that we all have. So after the presentation is done, if we're out of time available for questions, the speaker can then continue to dialogue with the rest of us on any remaining questions. So please feel free to use that chat. So we're going to go ahead and get started then. And Dr. Becker comes to us from Montana State University. He's going to talk about movement screening to prevent musculoskeletal injuries in adolescent runners. Go ahead, Jim.
Great, thanks, Curtis. Thank you for that introduction. So running, and in particular participation in track and field and cross country, is the largest participation sport at the adolescent and high school, middle and high school levels. And in Montana in particular, these 2 sports account for 25% of all sport participation. There have been numerous studies that show that running in particular has great benefits, including improved cardiovascular function, decreased obesity, increases in self-esteem, and decreased depression symptoms, right? So all of those are great things. The problem is the injury rate. Several epidemiology studies looking at almost 3,000 middle and high school runners found that up to 68% of these individuals reported having sustained a musculoskeletal injury in any one previous year period. We know these injuries have social, economic, and emotional costs associated with them. We know that the best predictor for identifying an individual who might sustain a running injury is have they had a previous injury. Right? So there's a lot of these secondary injuries occurring. And when we look at these secondary injuries, we see there's a 4-fold increase in the number of injuries that are season-ending or that ultimately result in medical discharge or disqualification from further sport participation. If that happens, we see a dramatic increase in worrisome conditions such as increased obesity promotion behaviors, decreases in self-esteem, declines in mental health, and these can have long-term impacts on overall physical activity and health patterns. And so we really want at an early age to try to prevent these injuries from occurring. In order to do that, we need to understand what are the injuries and how do they occur in the first place. And so if we look at both the middle and high school levels, we see that the common running injuries are mostly soft tissue, a little bit more bone stress injury at the high school level with running higher mileage. But the thing that all of these injuries have in common is they are all chronic overuse injuries, right? Which means that they are resulting from repetitive applications of load, that is either beyond the athlete's ability to handle at that time point, or is being applied too frequently with not enough recovery in between sessions. And so if we want to understand at an individual athlete level, what are the loads their tissues are experiencing, how likely are these loads to potentially cause injury in them, we have to look at how they move and what are the loads in their tissues. In a biomechanics lab, this is relatively easy to do. We can bring our athletes in, we can cover them with reflective markers, run under motion capture on an instrument treadmill. We can measure all the details about how they move and what are the forces applied to their body. Most adolescent runners, however, don't have access to a fully equipped biomechanics lab. For example, in all of Montana, there are 3 such facilities that could do that kind of analysis. So we have to come up with alternative strategies to get a sense for what are the loads that athletes experience. And so one thing we could potentially use are movement screens. So movement screens are designed to have athletes control their body movement while we place them in positions that challenge their flexibility, their range of motion, and their overall control of movement. So one common screen that we use a lot is the single limb step down, shown here. You can see the athlete has to control the lowering of their body as they lower themselves off this box, trying not to drop the hip, trying not to let the knee collapse in, let the foot collapse. There have been numerous studies that have shown that injured runners perform worse than healthy runners on this single limb step down. Another common screen that we use is called the Y balance test. And in this test, the athletes have to balance on one foot while reaching as far as they can in an anterior, posterior, lateral, and posterior medial direction. Again, while trying to keep that leg nice and straight, not collapse in immediately on that support limb. And there's been numerous studies that have shown that the Y balance test is prospectively predictive of who will subsequently develop injury. And so our overall goals for this project, for our first aim is we want to know, do performance on these 2 movement screens provide us insight with regarding the loads that the athletes experience at common injury sites while they're running? And then secondary, to follow that up prospectively, to see can performance on these screens be used to identify runners who then subsequently get injured before that injury occurs, so we have an opportunity to intervene. The final question we need to ask here, though, is, okay, who should administer these screens? In an ideal world, this is a great place for a certified athletic trainer. These are medical professionals, usually the first person that an injured athlete would interact with. They are trained to screen for injury. They are experts in rehabilitating and designing prevention programs. The problem comes when we look nationally at how many schools have athletic trainers on staff. We see that only 37% of public high schools in the country have a full-time certified athletic trainer on staff. And then in the Mountain West region, we're doing substantially worse, right? Montana, only 9% of schools. Idaho, Nevada, 17%, Wyoming, 24%, Alaska, 0% of schools have a full-time certified athletic trainer. And so we have to find strategies to be able to use these movement screens without having to rely on having these medical professionals present in the schools, because they may not be there. And so the third aim of our overall project is to see, like, can we teach coaches and parents or other non-healthcare specialists how to perform and reliably and validly assess these movement screens. And so that kind of gives an overview of this whole project that we're working on. For the rest of the presentation, I'll kind of just focus in on this first aim and look at some of the data analysis that we've done regarding whether performance on the single limb step down does actually tell us about the tissue loads these adolescent runners experience. And so far, we've had 35 runners participate in our study. If you look at our age 12 plus or minus a year, so we're really focusing kind of on the middle school aged runners. But just because they're young doesn't mean they are not accomplished. These individuals are running on average 24 miles a week, and they're quite fast, 5 minutes, 40 seconds average personal best for a mile. So these are really accomplished young runners. So they come into our lab, we cover them with motion capture markers. They run on an instrumented treadmill. We give them 5 minutes to warm up at a self-selected pace. to get used to running in the lab environment with all the markers on. And then we have them run at whatever their easy, regular training run pace is while we collect 3D motion capture of their motion and use the instrument treadmill to measure the forces being applied to their foot. From this data, we can then go ahead and actually calculate the loads at common injury sites. And so for this study, we're going to focus in on the Achilles tendon and the patellofemoral joint, as those are two of the most common overuse injury sites. And so at the Achilles tendon, if we know the joint torque at the ankle calculated from the motion capture data, and we know the joint angles also calculated from motion capture data, we can then fit those joint angles to some regression equations from the literature to derive a lever arm for the Achilles tendon. And if we think back to physics, right, if I know the torque and I know the lever arm, I can then calculate the force that is producing that torque. So in this case, the force in the Achilles tendon. So that's our first variable that we quantify. And then we look at several aspects of that force, including the peak force the athletes experience during stance, the loading rate, or how fast is that force applied, the impulse, or the area under that curve on each step that they experience, and then the cumulative or total load that that tissue is going to experience per kilometer run. We do the same thing at the patellofemoral joint. It's just the knee is a little bit more complicated because we have to account for co-contraction of all the muscles that cross the knee joint. But from our motion capture data, we can calculate the forces in the hamstring and gastrocnemius, figure out, okay, how much flexion torque are those creating at the knee, add that to a knee extensor moment, we calculate from inverse dynamics, and eventually we solve for the force in the quadriceps muscles, and then a certain percentage of that force is actually causing a compressive force at the patellofemoral joint. We can use MRI studies from the literature to look at the contact area between the patella and the femur. And if we know the contact force and the contact area, we can then calculate the stress that joint is experiencing. And the joint stress at the patellofemoral joint is one of the main determinants of patellofemoral pain syndrome, who would develop that. Just like at the Achilles, we have several different metrics we look at, including the peak joint stress, the rate at which that joint stress is applied, the impulse, or the area under the curve on each individual step, and then the cumulative impulse that the athlete experiences at the patellofemoral joint per kilometer run. For our single limb step down, we had the athletes perform 10 consecutive repetitions. And again, we use the motion capture data to quantify measures such as the amount the pelvis drops, the amount the hip adducts or internally rotates, how much does the tibia rotate, how much does the rear foot collapse. Greater values or greater motion on all of these things is indicative of worse performance on that single limb step down. So for our data analysis, since we have a bunch of predictor variables and a bunch of outcome variables, we start by using a principal component analysis, where we take all of our variables for the Achilles tendon loads, all of our variables for the patellofemoral loads, all of our variables for the single limb step down. We put them into a principal component analysis to look at the principal components extracted and then those factor scores. Since these factor scores are going to vary based on our sample, we then look at, okay, what are these factor scores representing based on the loadings to calculate surrogate variables. And from our lab, we've seen these are translatable across different samples of populations. And these surrogate variables should be highly correlated to the component factor scores if this analysis is done correctly. And so we always check that step as part of our results. And then we look at those surrogate scores. Do they actually predict the load in the tissue? And so our results for a single limb step down, we end up with two principal components that were extracted. One that represents frontal plane collapse during that movement. A second that represents rotational dysfunction during that movement in the transverse plane. So we see, do those predict the load in the Achilles tendon? And do those two surrogates predict the load in the patellofemoral joint? So in terms of our just descriptive statistics, these are our means and standard deviations for how much the athletes were moving during the single limb step down. They're all in normative ranges for what we would expect for the range of healthy athletes performing this movement. loading of the Achilles tendon, again, all within normative ranges. It is notable that the Achilles tendon in these young athletes, even at this age, up to 6 times body weight load going through that tendon while they're running. And similarly at the patellofemoral joint, all of our loading metrics were within what we consider normative ranges. We look at, did our surrogates actually capture what the principal component analysis produced? So for the Achilles tendon, we can see that all of our variables load very strongly onto one principal component. And so we calculate our surrogate for this as the average of all of these variables. And when we compare, does that surrogate actually represent what the principal component extracted? We can see we have a very strong correlation here. So this surrogate is accurately capturing our principal component. Same thing at the patellofemoral joint, except in this case, only 5 of our 6 variables were included in the analysis because our 6th variable didn't load very strongly onto that principal component. So in terms of our overall regressions, at the Achilles, it seemed like the single limb step down did not predict Achilles load across any of our participants. Our R-squared was only 0.02, so we're only able to account for 2% of the variance in Achilles tendon load from the single limb step down. However, we look at the patellofemoral joint, we fared much better. Athletes with worse medial collapse and worse rotational dysfunction on the single limb step down displayed greater patellofemoral loading while running. And both the frontal plane collapse and the rotational components were significant predictors within this regression model. And so our takeaway messages here. So it looks like runners who cannot control frontal plane movement or transverse plane rotation during the single limb step down are also experiencing greater patellofemoral joint loads while running. And this isn't terribly surprising, as similar combinations of movements, greater frontal plane motion, greater transverse plane motion, have been observed in runners who are currently injured with patellofemoral pain syndrome. But I think we might be able to apply this to other injuries as well, because these similar movements have been shown in runners who are symptomatic with things like IT band syndrome, other common overuse injuries. So the single limb step down might be a really good screening tool for injuries besides just patellofemoral pain syndrome. It did not seem to be really predictive of Achilles tendon loads. And so at this point, we don't think the single limb step down would be good for Achilles tendon injuries. That may just be due to the knee dominant nature of the movement. When we watched the athletes, a lot of them didn't use the ankle very much as they performed the single limb step down. So maybe if we use a screen that has much more ankle involvement, like the Y-balance test, we might be able to better predict athletes who have high Achilles tendon loads while running. So in terms of back to our original aims and where we are and how we're moving forward, so does performance on single limb step down provide insight regarding tissue loads? The answer appears to be yes. And so our next steps, we want to refine our musculoskeletal models to look at some of those other common injury sites, such as the IT band or the tibia for bone stress injuries. And then repeat this analysis using our second movement screen to see how the Y-balance test fares. So one screen might be better than another. Can these be used to prospectively identify runners who subsequently develop injury? Well, so far in our cohort of 35, we've only had 3 injuries, so we can't really answer this question. You always feel a little bad doing a study saying, oh, I hope people get injured, because we don't want them to actually get injured, but we don't have enough injuries yet to really answer this question. And then third, in terms of being able to do this with coaches or parents or other non-healthcare specialists, we are currently training a cohort of parents, and this spring we will be comparing their assessments of these movement screens to what the motion capture data actually said, to see how validly and how reliable non-healthcare specialists can assess these movement screens, to see if they could be used in a wide population that doesn't have access to athletic trainers or biomechanics labs. So with that, I'd like to just acknowledge the students that have been helping with this project, helping collect data and do some of the analysis, the athletes from the Bozeman Track Club for participating, and I'd be happy to answer any questions anybody might have.
Thanks, thanks, Jim. That was really great. There are a couple of questions in our chat, so let's start with that. So there's a question from Janice Bowie about what, if any, preventive strategies are being deployed to avoid or reduce injury before it occurs. I assume that means outside the context of your study. So like what's currently being done in these adolescent running populations.
It's really going to depend on the population that you're seeing. I don't know if there's any sort of standardized kind of thing that all running groups are doing. In our group, they do try to incorporate some basic strength training, some basic flexibility, but there's not a lot of formal organized injury prevention stuff being done.
Gotcha. And the question from Yemen Chin has to do with how do you incorporate, if at all, body composition, other factors of these subjects in your study into?
Yeah, body composition is a tough one to include. We do account for body weight. I mean, all of our variables, anything that's load related is normalized based on body mass. So the body masses of the participants get factored in. In terms of the body composition, we don't at this point have a good way to account for that.
Gotcha. A related question: how translatable would your approach be to other populations, say different age groups or different levels of baseline running skill, let's say?
Yeah, I mean, we've done kind of similar analyses in large cohorts of adult runners, and we see similar relationships in that performance on the single limb step down in particular does appear to provide a lot of information about how you're going to move while you're running. And so if you see a lot of this medial collapse or rotation in the step down, you also see those motions while the person's running. And so I think it does translate pretty well.
Gotcha. Well, great. I think, Jim, we're going to move on to our next topic, but I want to point your attention to the chat window. Evan Papa has a question in there. He's in your field, so it's a very technical question, and I'm hoping you can handle that via chat.
Sure.
Thanks, Jim.
Yeah.
So our next speaker, Chris von Bartel. Go ahead, Chris, and get your slides pulled up. Thanks for joining us today. Dr. von Bartel and his colleague Mitchell Strominger come to us from the University of Nevada, Reno. Looks like it's working okay, Chris.
Okay, can everybody hear me? And
yeah, we can hear you. Go ahead and
can you see my screen?
Yes, we can. You might want to put it in presentation mode, or if you like it like that, that's fine as well.
Well, I think I'll leave it like this and I can move the mouse around.
That's great. Yeah, fine. Thanks.
Okay, first of all, I want to thank the organizers for inviting me and for us and to present our work here. I want to introduce myself. So I'm the basic scientist in this team, and my clinician is Dr. Strominger, who is a clinician and ophthalmologist. I'm more of a cell biologist, neuroscientist, and with new interests in epidemiology. And so we got this DTTG grant, and that is a developmental translational team grant. And it's supposed to help us provide proof of concept so that we can get a bigger grant later on. And so in my next slide, I actually want to acknowledge and tell you that the CTR-IN, and that we really appreciate the funding from the CTR-IN and also from INBRE. And these small grants have really helped pave the way to funding of our new project. So some of this actually all the way goes back to when I had a pilot grant on strabismus and schizophrenia. Then we got the DTTG grant last year. And we've had INBRE service awards that have also helped with funding. And just a few months ago, we actually got an R21 grant funded. So we seem to be on the right track. So my next slide is a little bit, because not everybody might be familiar with what is strabismus? So strabismus is a deviation of the visual axis in the horizontal plane, horizontal strabismus. There's also vertical strabismus when the deviation is in the vertical direction, but that's relatively rare. More than 90% of all of the strabismus is horizontal. There are two forms. The eye can either be inward deviated, as shown here, and we call that esotropia, or the eye can be deviated outwards, and that's called exotropia. And this is not just a cosmetic problem, as somebody might think, but if this happens, as usually it does in early childhood, during the time when the binocular neurons develop in the cortex, this can really prevent binocular vision from forming, and it can be as bad as that, because the cortex doesn't like it when it gets images that cannot be fused. So it will shut off the information that comes from the weaker eye, and then you might actually lose vision in your weaker eye. That's called amblyopia or cortical blindness. So this is not a benign condition. This is something that, especially in childhood, you need to pay attention to. So what is the global prevalence of strabismus? You can find studies that say that it's 0.1% or studies that say it's 10%. Usually you find quotes of 2 to 4%. But when we needed this information, what is the global prevalence of strabismus? When I tried to find a comprehensive review, I was pretty much shocked that it didn't exist. And so then I actually started, how difficult can it be, right? To compile all the information that I could on studies that reported about esotropia, exotropia worldwide. In 2019, a study actually appeared that claimed that they did a global review, but it was only based on 28 studies. And in the meantime, I had already found nearly 300 studies. So there's a lot more out there than, and when we compiled all this information, we were pretty surprised how variable the patterns are between esotropia and exotropia, the two forms of horizontal strabismus. And when you compile that, you can see here that the esotropia prevalence is in blue and the exotropia prevalence is in red. And you can see that each ethnicity has pretty much its own signature of, and there's these big differences from one ethnicity to another. And only 2 or 3 of them had actually been described before. So we were really, by putting all these little puzzles, like a jigsaw puzzle, once you have enough of these little pieces assembled, you start to see the patterns. And so we can actually make these global maps of which ethnicity has what kind of horizontal strabismus pattern. And so to me, it was really eye-opening when I realized that Native Americans have pretty much the same pattern as East Asians. And of course, we know that Native Americans 10 to 20,000 years ago moved from Central Asia to the Americas. And so they kept, for these tens of thousands of years, their pattern, which kind of pointed to that whatever was responsible for these differences between ethnicities must go back tens of thousands of years. So we kind of became interested in not only human migrations, but also in where do these patterns come from. And tens of thousands of years ago, actually there was interbreeding between more archaic human cousins of humans like Neanderthals and anatomically modern humans. And so we looked into some of the literature on Neanderthal gene percentage in ethnicities. And when we plotted that against the esotropia-exotropia ratio, whether you had more esotropia or more exotropia, you can see that the more Neanderthal genes you have, it seemed like it was a pretty good association between that and having more exotropia. So we started to think about what was actually going on, so it seemed that probably the orbit itself was contributing maybe to these different patterns of strabismus. So there had been a little bit in the older literature that was proposing that people with a wider orbit and a larger interpupillary distance might have more exotropia, but that was never really formally evaluated. Now, if you think about it, Neanderthals had larger heads and a wider orbit, so they would have had a wider interpupillary distance than anatomically modern humans. So when Neanderthals and humans interbred, could the Neanderthal gene integration in humans cause a wider orbit? And that might be in some populations more than in others. So what is known about how is the width of the orbit controlled? That actually has to do with the width of the frontal bone. And so these osteoblasts, they keep dividing and dividing, and they require a signal to actually stop dividing and differentiating. And that's done by two transcription factors, RUNX2 and TWIST1, especially RUNX2. And the timing of that obviously is important because later this transcription factor is activated, the wider the orbit will be, and so that would be expected to be different in Neanderthals than in humans. So we then, so I started to pay a lot of attention to some of the work that had been done on the Neanderthal introgression in the human genome, and one of the main people in that field is Josh Achilles, who's now at Princeton, and he had published about Neanderthal introgression in more than 1,000 human genomes from many different ethnicities. And so I sent him an email and asked if he would be interested in collaborating on the project. And then he said, well, here's my primary data, and I think what you want to know, you can find out by looking at these primary data. So I looked up his files, and they were all bed files. I had no idea what that actually was, but so I consulted with our bioinformatics core, Julie Peterwright. Not only knew how to open them, but also how to interrogate them. And I asked her to look at about 30 orbit development genes, and asked if she could find out which ones had how much Neanderthal introgression. And that was pretty amazing. That was about a couple of years ago, because she came up with two big hits, and it was RUNX2 and TWIST1, exactly what are the key regulators of the that are responsible for the width of the orbit. And so now Josh Achilles' data were a little bit, they were not very precise in telling us where these changes were in the Neanderthal SNPs, the single nucleotide polymorphisms. They only were looking at larger pieces of DNA and which ones had more Neanderthal components. So we needed to do some remapping, and Hans Vasquez-Gross from the Bioinformatics Center at our university helped us then with the mapping of the Neanderthal SNPs in the RUNX2 gene. So there was a lot of introgression though, and there were basically two different patterns that we found. And none of these actually affect the protein coding region. The RUNX2 transcription factor, the protein, is exactly the same between Neanderthals and anatomically modern humans. But where the difference are in the regulatory regions of the gene. And so we were able to, taking advantage of the preliminary data that we had accumulated on the ethnicities and how many of them had more exotropia and more esotropia, the region that seemed to be most interesting was the one that is colored here in orange. And so once we got the information about all the where the SNPs were in the RUNX2 gene, we then could narrow down which parts we wanted to actually interrogate. So the research question of our DTTG grant was to test the hypothesis that Neanderthal introgression in RUNX2 contributes to a wider orbit and therefore more exotropia. And if this was true, then we would expect to see more RUNX2 Neanderthal SNPs in people that have exotropia than in people that have esotropia. And so the research plan was pretty straightforward to, first of all, identify the sites of the Neanderthal SNPs in RUNX2, exactly where they were, and then design the suitable primers for these regions, extract and amplify DNA from patients that had either esotropia or exotropia, and then sequence these regions and look at how frequent they were in this regulatory region of RUNX2. So, as I already mentioned, Hans Vasquez Gross from our bioinformatics center was really great in not only helping us with the mapping of these SNPs in RUNX2, but also in the design of primers. So that helped a lot. And then we had from previous studies in our freezer de-identify DNA samples from patients that had either esotropia or exotropia. We actually had 2 ethnicities initially. We were able to add a 3rd ethnicity from South Asia because I had an international scholar join the lab in the meantime. So we had actually 3 groups of samples that we could do our experiments on. And then I had people in the lab, my international scholar and technician, who did all the lab work, DNA extraction, PCR amplifying, and sequencing was done at the Genomics Center. So we here, the next slide.
About a minute to go, Chris.
Run out of time. Okay, I'm almost done here. This is really fast. So we had 20 samples for exotropia and 22 samples for esotropia. And you can see here as green coded, those that had, and 3 of the 7 regions that we so far tested had, they were, had the Neanderthal SNP in both alleles. And there were 3 out of the 20 for the exotropia samples and 0 out of 22 for the esotropia samples. And so this is just highlighted here in the next slide. So we had, when you do the quantification, we had 15% of our exotropia samples had Neanderthal SNPs in these regions that we think now are responsible for a wider orbital width. And this is just to acknowledge the help of the people that I already talked about and the funding that we received for the project. Sorry, I went over. I thought I could present this in not fast.
Yeah, you're about spot on time now. That's
okay.
great. Yeah, thanks.
Do we still have time for questions or?
We do. So
Okay.
yeah, so I'd love to entertain some questions. I'm not seeing anything in the chat, but if you've got a question, please just unmute and chime in.
Okay, I was typing, but I'll ask
Go ahead.
my question verbally. This is very interesting. What I'm curious about is looking at SNPs individually and inferring Neanderthal introgression from them. I mean, we can do that, versus looking at haplotype effects and assigning local ancestry to the entire segment. And so do you have any data to suggest that it's the entire RUNX2 gene that's descended from the Neanderthal, or whether it's just the acquisition of these variants over time through different injections through the intermarriage in the gene pool? I know that's a real technical question. Sorry to.
Yeah, I don't really have the answer to that. My understanding is that, well, there have been multiple interbreeding events between Neanderthals and humans. And so all South Asians, East Asians, and Caucasians actually have can have rungs 2 from Neanderthals.
Yeah.
When we were looking at twist 1, it was actually different. Only East Asians have rungs 2 that has Neanderthal introgression. So there are some differences between ethnicity, and that kind of makes sense because the East Asians had a more recent interbreeding event with Neanderthals than South Asians and Caucasians. So there might be differences between ethnicities in that respect, but I cannot really answer your question.
Yeah, and it would just be interesting to know whether it's variation in the gene and it's the Neanderthal introgression that clued you into it, but the alleles don't have to be from Neanderthal ancestry. It could just be a variation in this particular gene versus variants that must be from Neanderthal introgression. And that leads to different hypotheses about the mechanism involved. So I think this is really interesting work. Congratulations.
Well, thanks. Yeah, we have to get consultations with geneticists on this once we have a little bit more data and really write the manuscript. And so.
Well, Josh Akey is certainly a really good one to be talking to. So
yeah. I will try to get him involved once we have firmer data. This is all kind of a developing story. Thanks.
Thanks, Chris. Any other questions? We've got about a minute if there are other burning questions.
I have one more comment, and that is, so
Sure.
this might be relevant beyond just ophthalmology and strabismus, because if you think about, and this is a segue into the next talk, which is on cognition. So when you look at what clinical or psychological cognition people, they show these images, and please ignore that one of them is smiling, one is not. But which one is introvert, which one is extrovert? It's pretty clear. This one looks more, you would say this one is more introvert, this one more extrovert. And if you look at the interpupillary distance, this one actually has 10% larger interpupillary distance, wider orbit. So if we really have identified these polymorphism region that is responsible for that, this might be relevant beyond just strabismus ophthalmology, but it might actually change cognition. Just some food for thought.
Interesting. Well, thank you, Chris. That was a really fascinating talk. You covered a lot of territory in a short period of time, so we appreciate that.
Okay, thanks.
So our next speaker is Shi Chin from University of Idaho, and if you want to go ahead and pull up your slides.
Sure.
So while she's getting that going, she'll be talking to us. Dr. Chen will be talking about determining the impact of emotive intelligence space on children's self-regulation and cognitive performance.
Okay. Can you guys see my slides okay?
Yep, looks great.
Okay, perfect. Yeah, my name is Shi Chen. I am an assistant professor at Margaret Ritchie School of Family and Consumer Sciences at the University of Idaho. And our project is determining the impact of emotive intelligence spaces on children's self-regulation and cognitive performance. And our project is led by a team of multidisciplinary researchers. I am the PI on this project. My background is in educational psychology, and I was joined by my colleague at the University of Idaho, Dr. Lin Hao. Her background is child development. And this is also a collaboration between University of Idaho and Washington State University. My colleague Mona Gandhi and Mingyang Sarudi are faculty at Department of Design and Construction at Washington State University. Some other key personnel, including Dr. Hassan Hasdan. He is an associate professor of computer science at Washington State University. And my right-hand person, my graduate research assistant, Rebecca Cimino, and my undergraduate research assistant, Ashley Jaleef. They are instrumental to the data collection of this project. The purpose of our project is to determine the effect of emotive intelligence spaces, EIS for short, on children's behavioral, cognitive, and physiological outcomes. I will get more into what EIS is in later slides. In short, EIS is an intervention mechanism we created that leverages human color perception and affective computing. And this is actually the second phase of a larger study. Phase 1 was completed about 2 years ago. During phase 1, the affective computing algorithm was created based on physiological data and emotional data collected from college-aged adults. And in phase 2, children's data were collected to calibrate the algorithm and to determine the effect of EIS on children's physiological and cognitive outcomes. And in phase 3, which will start this fall, EIS will be used with children with ADHD to investigate its impact on attention and self-regulation. Some background information, we choose to study the population in rural area of Idaho because living in rural area is associated with a number of risk factors. For example, lower house income, lower parental education, limited access to quality healthcare, poor academic achievement, and poor self-regulations. And in Idaho, Idaho has a higher percentage of people living in rural area than the US average. And we chose to study self-regulation is because self-regulation is a set of ability or cognitive gadgets, if it will, that underpins a lot of future academic and cognitive and mental health outcomes. And a lot of study has shown that there's a strong association between early self-regulation and future mental health outcomes, cognitive performance, inhibitory control, depression, anxiety, and academic achievements. Self-regulation is the ability to manage emotion and impulses. And we choose to study very young children is because during this age, this is a very sensitive period to nurture brain circuitry related to self-regulation. And children this young, their prefrontal cortex, where self-regulation is believed to occur, is far from maturing, which means that they have very limited self-regulation ability during this stage. And that underlines the need for co-regulation. Co-regulation is to cultivate self-regulation with environmental assistance. And therefore, we had this idea to create our intervention mechanism. We call it emotive intelligence spaces, or EIS for short. This is what a prototype EIS looked like. EIS consists of 6 wooden wall panel with interactive modules with LED lights. The 3 light thingy you can see on top, those are the lights, creating a 90-degree semi-private spaces. The colored environment is set based on participants' emotional states. The lights are controlled by an app on our phone, which also monitors children's physiological reactions via a medical grade wristband. This is what an EIS looks like when the lights are on. We program several different colors based on children's preference, and when we collect data, we usually dimmer the lights so to create a more immersive environment. The EIS design principles are these 2 theories. First is your Adolescent law, stating that an optimal level of performance is associated with an intermediate level of environmental arousal. And the second is based on attention restoration theory, stating that individuals' ability to direct attention is supported by environmental features that elicit cognitive restoration. And for our study, we wanted to investigate through research question. We want to know whether being in the EIS environment has an impact on children's self-regulation, working memory as an indicator of their cognitive performance, as well as their physiological reactions, such as heartbeat, galvanic skin responses, and skin temperature, controlling for age. And children's physiological data was collected using Empatica E4 wristband. This is a medical grade wristband with sensor on the back. It collects data such as blood volume, pulse, and skin temperature. And for children's working memory, we use Wilcox-Johnson 4th edition cognitive battery number reverse subset. This is a very well validated and widely used cognitive battery. And we choose this test because it's age appropriate and it's very easy to administer. In this test, the experimenter will say a random combination of numbers, and children will repeat those numbers backwards to the experimenter. For children's self-regulation, we use the head, toes, knees, and shoulder test developed by a lab at Oregon State University. This is a nonverbal recall task where the experimenter, just like the game or the song, the experimenter will say different combinations of head and shoulder, knees and toe, and switch game rules. And children will follow direction and do the opposite of what the experimenter is doing. We use an ABAB design to test the effect of BIS. And this chart illustrates our experimental design. Each baseline and intervention condition lasts about 5 to 7 minutes, and we have